Welcome to another Anatomy and Physiology II Bio 265 video in a nutshell. Today we are going to be talking about the Clotting Cascade. The purpose of the Clotting Cascade is to ultimately stop bleeding, and it starts with proteins that are mostly produced by the liver, and they circulate in the bloodstream until there is some kind of damage, and then they activate in order to basically form a scab, tie everything up. There are two ways to start the Clotting Cascade. The first is known as the intrinsic pathway, and the second is known as the extrinsic pathway. And we'll talk about what those names mean later on. The intrinsic pathway starts when the cut exposes collagen fibers that are typically covered up by endothelial cells. So this intrinsic pathway is started the exact same way that the platelet plug is. Collagen fibers that are going to bind to a protein. Platelet plug, it's BWF. Here, it's going to be something called factor 12. So factor 12 is going to, when it meets collagen, it's going to be converted into 12A. And then factor 12A is going to bump into factor 11 and turn it into factor 11A. And 11A, get the process, is going to bump into 9 and turn it into 9A. 9A can't act all on its own. It needs 8A. And we don't talk about in this class what does the conversion, so don't worry about it. Just know that we have 9A and 8A, and the two of these combine together in order to convert 10 into 10A. So we've gone through, we've done these conversions, we've reached our 10A. This is the intrinsic to reach 10A. That was our goal. Now let's jump over to the extrinsic. And the difference between these, remember how we talked about the intrinsic being collagen fibers that are right at the level of the blood vessel. One way to think about or understand the use of the word extrinsic, let's suppose that right here is our bloodstream, and up here on top is the surface of our skin, and right here is the cut that comes on down through and reaches the blood vessel. So we're going to have blood that is going to be traveling up and spurting out up through here. There are cells here that line the edges of this cut, and all of these cells here on the side have been damaged. Now, down here at the level of the blood vessel, these little things here might represent the exposed collagen fibers. So these exposed collagen fibers down here, right at the level of the blood vessel, are going to set off the intrinsic pathway. Now these cells out here in the surrounding areas, they have also been damaged, but they aren't right at the level. But inside of them, they have tons of little proteins that are called tissue factor. And so when they get hurt, they kind of say, hey, we want to jump in too, we want to help. And so they are going to release this tissue factor out into the wound. And this tissue factor, it's going to float down, and it's going to help start the extrinsic pathway. So now jump over to the extrinsic pathway. Now you can see why these ones are extrinsic or further away from, and these ones are intrinsic or closer to. So like we said, the extrinsic pathway is going to start with a protein called tissue factor. And when tissue factor gets involved, it is going to grab factor 7 and turn factor 7 into factor 7A. And 7A, it's actually going to be very direct because 7A can act directly on 10 to turn 10 into 10A. Notice how much quicker this extrinsic pathway is. But the net result of both of them is that we turn 10 into 10A. So we call this conversion of 10 to 10A the common pathway from here on down, because up here it's separate, and from 10 to 10A, they meet. So 10A, it can't work all on its own. In the same way that 9A and 8A had to combine, 10A is going to have to combine with 5A. And don't stress about what does the conversion of 5 to 5A, you don't need to know that. But just know that we have 10 and 10A that are going to team up, and they are going to convert prothrombin to thrombin. So we have this conversion completed, and then thrombin is going to act on a protein called fibrinogen and turn it into fibrin. Now, if you remember, we talked about fibrinogen previously during the platelet plug. Fibrinogen hooked platelets onto platelets. Now you find that it can be converted into something new, into fibrin, and have even a different function. So this fibrin that we've created, it looks kind of like little tiny strands of protein, and those fibrin strands hook together kind of like this into a big, long, basically it's kind of like thread. And that thread is going to meet up with another factor called 13A. And 13A is kind of like the needle. And 13A is going to take this fibrin thread and it's going to sew it through the platelet plug. So if you understand the platelet plug, we have all these platelets that are hooked together and things piled on top of them and red blood cells, and we kind of have this big jam of things that are stopping the blood flow. but the platelet plug just isn't long-term, it'll fall apart. So these fibrin strands along with the 13A needle come in and they sew between all of those platelets and red blood cells and proteins and whatever else is piled inside of that platelet plug, and it's going to sew it together tight and lock it in. And so then as it dries, you're going to get a solid scab that doesn't fall apart because it's sewn together now. And if you're ever curious, you can take a scab, and if you take a scab and pull it apart, it sounds kind of gross, but you can see the little fibrin strands breaking inside of that scab. Now, in order to break this all down, so now we've created our clot, we have something called plasminogen that can be converted into plasmin. And plasmin is the cellular equivalent of a pair of scissors. And those scissors are able to come and cut the thread that was formed with the fibrin strands. So when plasmin is present, it's going to come chop up those strands and will recycle the clot. And the way I convert plasminogen into plasmin in order to get my scissors is I use something called TPA, or tissue plasminogen activator, and tissue plasminogen activator converts plasminogen into plasmin. So for those of you who are going into medicine, if you give someone TPA that is having an ischemic stroke, you are converting their plasminogen into plasmin, and you are creating the scissors that are going to go in and cut apart the clot that's giving them their ischemic stroke. Thank you, and good luck with your studies.